Our story begins with a young, 1-year-old, domestic short-hair cat who was brought to a veterinary specialty hospital with a perplexing set of symptoms. This wasn't just a case of a cat being a bit clumsy. This feline was experiencing progressive difficulty walking, a condition that had been worsening since it was 6 months old. Imagine a cat, once agile and graceful, now struggling with every step. But that wasn't all. The cat also had macroglossia, which is an abnormally large tongue, and dysphagia, or difficulty swallowing. The cat's tongue was so enlarged that it constantly protruded from its mouth, causing excessive drooling. On top of that, the cat's neck and shoulders were noticeably thickened and rigid, adding to its discomfort and limited movement. It was clear that something was seriously wrong, and the veterinarians suspected a congenital neuromuscular disease. The muscle biopsies confirmed the veterinarian's fears. The cat was suffering from a degenerative and regenerative myopathy, a disease of the muscle. This meant that the muscle tissue was breaking down and attempting to repair itself, a hallmark of muscular dystrophy. To pinpoint the specific type of muscular dystrophy, the researchers used immunofluorescent staining to look for the presence or absence of key proteins associated with the condition. What they found was telling. The dystrophic muscle showed no signs of staining for dystrophin, a crucial protein that provides structural support to muscle fibers. Specifically, antibodies targeting the rod and the C-terminus of dystrophin failed to bind to the muscle tissue, indicating a deficiency in this protein. In contrast, staining for utrophin, a protein similar to dystrophin, was increased in the dystrophic muscle, suggesting the body was trying to compensate for the lack of dystrophin. While the gene Titin, TTN, has been associated with MDs, the clinical, pathological, and histological data in this case supports a dystrophin deficiency, making further investigation of other genes less warranted. The young age of the affected male cat, along with the markedly increased CK activity, dystrophic phenotype in the muscle biopsy sample, and non-detectable staining for the rod and carboxy terminus of dystrophin with increased utrophin expression, is consistent with the diagnosis of X-linked MD. Muscle hypertrophy, a consistent finding in X-linked dystrophin-deficient cats, also occurs in Duchenne MD in humans and in the golden retriever model. This hypertrophy is considered true hypertrophy, not pseudohypertrophy related to fatty and connective tissue infiltration or hyperplasia. In conclusion, the expected LOF variant effect, population data, genetic data compared to humans, and amino acid conservation all support the identified variant as pathogenic and causing the clinical signs of X-linked MD in this cat. Precision/genomic medicine efforts for the domestic cat and in veterinary medicine increasingly support disease variant and animal model discovery, providing opportunities for targeted treatments for companion animals.